Our next speaker is from Mexico. Dr. Patricia Vélez. Can you hear me?
Loud and clear.
Yeah, thank you. Thank you. Thank you. Thank you. Madam, thank you very much for accepting our invitation to talk today. Welcome to the international online seminar on marine fungi. And we are very much excited to listen to you and your work on deep sea fungi. Thank you very much, especially for taking the time on a Sunday early morning, 7:30, 6. I think I saw you at 6 a.m. Mexico time here joining us. Thank you very much for that. Before I hand over this platform to you, I request Dr. Rohit Sharma, Managing Editor of MycoIndia Academic, to introduce you to today's audience. Dr. Rohit Sharma, please.
Thank you, Dr. Shenoy. We welcome Dr. Vélez to today's session of international seminar on marine fungi. So Dr. Vélez has completed her doctoral studies in biological sciences from National Autonomous University of Mexico for her work on Aeronicolous marine fungi. She performed 2 postdoctoral stays at National Autonomous University of Mexico and the Center for Scientific Research and Higher Education at Ensenada, studying molecular ecology of freshwater and deep sea fungi respectively. She is a faculty member, a research professor at Institute of Biology at the National Autonomous University of Mexico, where her work aims to explore the diversity, ecology, and potential utilization of fungal communities, particularly in marine ecosystems. She teaches courses on molecular ecology of microcommunities and taxonomy and conservation in Graduate School of Biological Sciences, and has mentored more than 20 graduate and undergraduate students in diverse topics of mycology. We welcome Dr. Patricia Vélez. So the floor is all yours. Please go ahead with your talk.
Thank you very much. Do you hear me?
Yeah, you are audible.
Yeah, excellent. Well, let me share. Okay, so I think you should be able to see my screen.
Dr. Vélez, please go to your desktop. Minimize this window.
Okay.
Go to PowerPoint or PDF or wherever you are presenting from.
Okay. Are you able to see my presentation now?
So this is PowerPoint. Can you go to the full presentation mode? Yes. So can you just go to the next slide just to check it actually? Yeah. Okay. Right. Thank you.
Okay.
Yeah. Please go ahead. Yeah.
No.
Thank you.
Well, thank you very much for the invitation. I'm really honored of being here. And well, good afternoon, India. Good morning, Mexico. And today I'm going to share with you some of our results on the exploration of fungal diversity in the deep sea of the Eastern Pacific Ocean. So the presentation is organized as follows. I'll begin with a brief introduction into oceans, the importance of these ecosystems. And also, we're going to briefly discuss some of the threats it is facing right now. Next, we are going to move into basic definitions and notions of marine fungi and deep sea ecosystems in Mexico. And the next points are going to be related with the results we've obtained so far on fungal diversity, some interesting remarks on deep sea hydrothermal vents and oxygen minimum zones, functional roles of fungi, metabolism of hydrocarbons, antimicrobial activity, and lastly, we're going to wrap up some conclusions. So as we may all here be aware of, the oceans are the lifeblood of planet Earth. and humankind. They flow over 3/4 of our planet, and they hold around 97% of the water, producing half of the oxygen in the atmosphere. But also they represent important carbon oxide sinks, and they account for nearly 61% of the global total gross national product. But they also play a very important role in the control of global temperature and weather. And well, this is very important for those nations or those countries that have coastlines or have or are island nations. So the sustainable utilization and conservation of the oceans is very important for the economic growth and food security of the entire world. However, the oceans are currently facing several threats originating from anthropogenic activities. So the combination of factors that are threatening a new mass extinction event in the oceans. Here I list some examples such as acidification, overexploitation, pollution, and sometimes this pollution leads to hypoxia, such as some oxygen minimum zones, and also very importantly, the climate change. So the joint effect of these impacts in the ocean, we've been, from science, we've been repeatedly evidencing that this is higher than we estimated in past years. However, this is still poorly understood. So here I'm sharing you some images of the last greatest oil spill in the Gulf of Mexico. You might be aware of the Deepwater Horizon oil platform. So baseline data is very needed on underappreciated species. Of course, charismatic species are very important. However, underappreciated species, such as fungi, represent the the majority of the marine biodiversity at both taxonomic and genetic levels. They are also widely distributed, and we do not know and have not reached a good understanding on the response to stressors of these organisms. So marine fungi, as I'm sure you already discussed several of these topics, are distributed across all marine ecosystems, from sandy beaches to coral reefs, mangroves, several coastal ecosystems, the water column here, but they also distribute in the deep sea. So first of all, we're going to quickly check the definition of a marine fungus. So this taxonomically diverse group of microorganisms have evolved and adapted to grow and sporulate. I mean, complete their entire life cycles under marine conditions. They usually occur as saprobes. However, also several pathogenic species have been reported, and they play very important roles in nutrient cycling. Despite all of these importances and the remarkable diversity, they represent less than 1% of the overall fungal described taxa. These microorganisms remain poorly understood, despite they inhabit fragile systems, such as sandy beaches or oil drilling areas. So we've already know from the literature that these organisms are susceptible to anthropogenic pressures. For example, they are susceptible to changes in the pH, the temperature, nutrient stability, salinity, and several other conditions. So we can expect that reductions in the diversity of these organisms may lead to changes at the functional levels of the ecosystems. So moving into the deep sea, the first isolation of deep sea fungus was from the Atlantic Ocean, from more than 4,000 meters below the sea surface. However, despite these early reports, presently the understanding of deep sea fungal diversity remains largely unknown. Of course, we know several terrestrial or terrestrial-derived taxa that may be acting as saprobes, and they are mainly associated to the marine snow, which is, I included here a couple of images so you can understand why this precipitation of nutrients from the upper water column is named marine snow, because it resembles actually what snow looks like. So, well, for this presentation, the definition of deep sea is delimited to those depths higher than 500 meters. So this ecosystem or this biome covers around 75% of the entire surface of the Earth. However, it represents the less explored ecosystem. It is largely cold, except for hydrothermal vents. It is always dark, and hence no primary production coming from photosynthesis is available. So this ecosystem is also oligotrophic, meaning a little nutrient ability is in this system. It has high hydrostatic pressure, around 1,000 times greater than on the surface, but it is very important in global carbon cycling and represents currently an alternative to mining on land. And also it's very important for the mitigation of anthropogenic climate change, because it plays a very important role in sequestrating carbon. And despite all of these importances and unique environmental characteristics, the biodiversity in deep seas is largely unseen, and so the benefits to human health and industrial activities. So the organisms that inhabit these ecosystems should, I'm here talking about fungi, should and most cope with extreme environmental conditions. So, well, here's a reminder of Mexico is located in the south part of the United States, and it has a very large coastline here, you can see, in the Pacific Ocean. And these marine systems include oxygen minimum zones, here represented as purple circles, but also hydrothermal vents, here represented as pink circles. So I'm going to talk about and share with you some of our results on these two ecosystems. But first, we need to define what are hydrothermal vents and what are the characteristics in oxygen minimum zones. So as I already mentioned before, the primary productivity of these deep sea ecosystems is not supported by photosynthesis. So here, microbial communities sustain life, and hence they drive ecosystems functions. So on the one hand, we have hydrothermal vents that rank among the 10 most productive systems on Earth, hosting an exceptional microbial diversity that is characterized by chemolithio-autotrophic bacteria and several heterotrophic microorganisms such as fungi. In the other hand, we have oxygen minimum zones that have a saturation level of oxygen that goes below the 10%. But despite this condition, they also host a rich microbial community characterized by anaerobic microbial processes that actually impact the productivity of the system and regulate climate balance. However, most of the investigations in microbiology in these two systems have historically focused in bacteria, neglecting microeukaryotes such as fungi. So, well, what we know from fungi in the deep sea, I mentioned it's a very few, but very important information is still available. So we know that the deep sea hosts endemic species. I included here the gorgeous images uh, from this paper from Nagano and Nagahama, describing an endemic deep sea fungus. We also know that the deep sea harbors a large diversity of potentially novel phylotypes of fungi that may be playing important roles, still unknown. And this is a curious remark, but very few studies have dealt with a culturable proportion of fungal communities. So this, of course, hampers the evaluation of ecological roles and the potential utilization of these bioresources. So, well, in terms of the potential applications of deep sea fungi, we know from the literature that the deep sea fungi have led to the discovery and isolation of several novel natural products, mostly with antibacterial and anti-cancer properties. They have been elucidated with the potential roles in buffering the impacts of the Anthropocene by having the ability to degrade or metabolize polymers such as plastics and also hydrocarbons. So overall, this evidence suggests that fungi are important in the utilization or degradation of plastics such as polyurethane. So we can only hope that if humans, we are unable to solve this environmental crisis on our own, fungi may eventually evolve to do it for us. So in this study, we sampled two different bend systems, three. The Pescadero Basin, which is white bands, the Pescadero Transform Fault, and the Alarcon Rise that are black bands. And also we included an oxygen minimum zone in the Alfonso Basin. All of these are in the Pacific Ocean, in the neighboring the Gulf of California. So we selected this methodology. First, sediment samples were collected on board the research vessel Western Flyer in collaboration with Embry Monterey Bay Aquarium in the US, using this remotely operated vehicle, which is equipped with robotic arms. After collection, the sediment cores were brought on board and subcores were obtained from each one of these. For fungal isolation, we subsampled from the center part of the sediment cores to avoid potential contamination. For culturing, we selected the direct plating method using different culturing media, and the obtained isolates were used to test for the their ecological properties, the potential role in bioremediation of hydrocarbons. Also, we conducted antimicrobial screenings, and finally, we conducted a bioactivity directed chemical study. But also, we implemented a complementary approach using the fungal metabar coding. This is a culture-independent approach. So in terms of diversity and ecological patterns, we observed very interesting things. Firstly, we identified around 100 ASVs. And here you can well, I'm not sure you can read all the names, but you can at least have a glimpse on the diversity. And very interesting, you can see inside of this red square, the large proportion of uncultured and unidentified, potentially new deep sea phylotypes that were, several of them were widely distributed across all the environments, all the sampling sites, and actually also occurring as a dominant component of the communities. Well, of course, Ascomycota here in red, and Basidiomycota here in blue represented the dominant components at the phylum level of the diversity. So in terms of beta diversity, and we detected also clustering patterns based on the ecological setting of the chimneys. Here you can see the white smokers that are characteristic of the Pescadero Basin here. And here, all the sites from Pescadero Basin cluster together. So this site is characterized by the large concentration of calcite, also the presence of aromatic hydrocarbons, and the dominance of this animal, the Osazicia alvini, which all of these are very different from the conditions that prevail in the Alarcon rice systems. Here you can see in purple represented, you can see how the site in the Alarcon rice is very different from the Pescadero. Here in the Alarcon rice, we have black smokers and the presence of polymetallic sulfides and the dominance of this tube worm, the Riftia. So it was very nice to see how these clustering patterns also resemble clustering patterns that have been reported from the benthic communities. So what I told you about the uncultured fungal phylotypes was also true in this other study conducted in the oxygen minimum zone of the Eastern Pacific. That is actually the largest oxygen minimum zone in the entire world. So here, what we sampled was a transect offshore the port of Mazatlan. And what I'm trying to share with you here is the large proportion of unidentified, potentially new uncultured fungal phylotypes that can only be determined as fungi SP based on the information available presently on databases. In this study from my student, Judith Posadas, a master's student, we also observed two interesting ecological patterns. First, the fungal diversity decreases with the depth. This means that offshore system sites have a less diversity than coastal systems, and this is reasonable considering the carbon input that these sites in the coast receive. And the second ecological pattern that we observed was that superficial samples here represented as dot zero have lowest diversity in comparison or in relation with the subsample portion of the same site, meaning that fungal diversity is higher beneath the surface in the seabed. This is something very interesting that is opposite to what has been observed in bacteria. You can see from these distances that superficial samples are super different from the subsurface samples. In terms of the culturomics, we isolated around 32 pure fungal isolates that clustered into 19 OTUs. All of them belonged to the Ascomycota, And well, here you can see some of the genera with a dominant presence of Aspergilli, Penicillium, Talaromyces, etc. The top abundant species, or OTU, was Aspergillus sidoid. This is very relevant. As you may know this fungus from several marine ecosystems. It is actually a true marine fungus, and it has been also related to the coral disease in the Caribbean and the pathogenic interaction with several deep sea organisms. So, well, these results, of course, are complementary to culture-independent approach because we did not identify this species from the metabarcoding data. So it is very good to see this. So using these isolates, we intended to understand the ecological relevance of these organisms in deep sea trophic chains. So for this objective, we used stable isotopes. This was some work done by my postdoc, Diana Salcedo. And what she observed here, you can see the red dots represent the Pescadero basin vents. The blue dots, the Pescadero transfer fault vents, and the green dots represent the Alarcon rise vents. So we observed that overall fungi are very important carbon reservoirs in deep sea vents. And they are playing very key roles in mobilizing carbon that is fixed via the chemosynthetic bacteria that I mentioned in previous slides. So you can see here, the first diagram is the results from the carbon isotope. You can see that around 60% of our isolates relied on the mixed carbon sources. This means from the photosynthesis that is precipitating from the upper water column and reaches the seabed. This is more less than 1% of the carbon actually. And 40% of our isolates relied exclusively on this metabolic pathway that is characteristic of vent systems. So this 40% of our isolates rely or are adapted to deep sea vents. And you can see here in the B graph, the nitrogen isotope results. So we can see here from that fungi are acting at different trophic levels, and they also depend on local and external nitrogen sources. And so they may be assimilating both bacteria and invertebrate necromass, functioning as parasites. And from this sulfur isotope, we were able to confirm that these isolates rely on chemosynthesis and epipelagic food sources. So here I want you to stick to the word bacterial, because it seems that the interaction between fungi and bacteria in deep sea vents is more important that we are aware of. So in order to explore this question, this is work done by an undergrad student, Blanca Martinez. We tested the interaction, the cross-kingdom interaction in a maze that she constructed. Here you can see the maze. So she inoculated the fungus in a single culture. She also tested the bacteria in a single culture, but next she co-cultured both fungus and bacteria together in the maze and observed differences in terms of solving the maze and dispersal capacities of these organisms when they are together. So, well, she did this experiment using these mazes with 5 different obstacles you can see here, including different difficulties. So, as a result, she observed that the interaction between the fungus and the bacteria is hitchhiking. You can see here the high phi, where bacteria seem or actually attached to the hyphae by the production of these polysaccharides. And at the macroscopic scale, you can observe this kind of pattern where bacteria get the benefit from the filamentous development of the fungus for dispersal. And well, this interaction seems to be detrimental for the fungus because it affects the growth and orientation of the hyphae. The fungus actually grows slower when the bacteria is present. So in order to understand what happens to the fungus when the bacteria is affecting its development during the interaction as observed here in the maize, we used a comparative transcriptome approach, and this was done by my master's student, Jessica Hernandez. So using this comparative transcriptomics, she also used this experimental approach using in vitro pairwise bioassays, where fungi, the fungus and the bacteria were co-cultured, and the controls were the fungus alone and the bacteria alone. So we observed the upregulation of transcripts that are related with the biosynthesis of antimicrobial compounds, endospore development, and biofilm production. All of this was upregulated in the bacteria when it is in the presence of the fungus. Here you can observe in the diagram how the upregulation of biofilm enhances the attachment of the cells to the hyphae. On the other hand, the fungus upregulates transcripts related with the production of mycotoxins and multi-drug transporters to cope with the results of this negative interaction. I forgot to mention here that the bacteria are producing also this we observed the upregulation of transcripts that are affecting the fungal cell wall. So perhaps bacteria are establishing this fear antagonism with the fungus, releasing the cellular components of the hyphae. So bacteria are both traveling and feeding on fungi when they are together. Of course, these are experiments that were tested under laboratory conditions, but still very interesting insights into the potential interactions between these two organisms. So, well, now in terms of the potential utilization of our isolates. So early evidence from the Gulf of Mexico and these two big major oil drilling regions, the Perdido and the Coatzacoalcos. We also obtained several fungi, also some yeasts, but we did not continue working with this. And we tested these organisms, you can see here, for their ability to use two different hydrocarbons, hexadecane and hexadecene, as sole carbon sources. And you can see here from the bar plot, the orange bars are the treatments where the fungus is, or the fungi are growing on the hydrocarbons versus the controls. So you can see how these fungi thrive on hydrocarbons. So it's very exciting results. So we decided to confirm this using a transcriptomic approach with this RNA-seq to identify differential gene expression. And we observed, well, actually we confirmed via the upregulation of transmembrane transporters, how the hydrocarbons entered the fungal cells. So we also detected several upregulated genes that are related to the metabolism, potential metabolism of these hydrocarbons inside the fungal cell. So with this preliminary results, we decided to use our vent isolates that, as you may remember, these two hydrothermal vents, one of them, the Pescadero Basin, has a natural presence of hydrocarbons. So we tested the fungal isolates. This is work that is being conducted by two of my postdocs. So we tested their abilities to use crude oil as soil carbon sources. Medium, heavy, and extra heavy oil, actually. So the results are very promising. As you can see here, this is a SATA HPLC graph showing the fractions, different fractions of extra heavy oil after fungal attack. So usually this higher, heavier fraction of the oil after fungal attack lowers, which means that the fungi are attacking these higher molecules, the carbons. It is very interesting because bacteria that are used for bioremediation avoid these heavier fractions of the oil. And I'm almost done. So yeah, I'll share with you now some of the results on the antimicrobial bioactivity of our isolates. And what we observed, the most exciting finding was that several of our species are targeting several pathogens. Here, I forgot, sorry, to explain that we tested the extracts of our fungi versus these pathogenic human pathogens. These are bacteria, and here we have a yeast. So we observed that our fungi target several pathogens, which is very promising. So also this suggests that this fungi in the deep sea, in the hydrothermal vents, are actually engaging in antagonic cross-kingdom interactions that, of course, may be affecting ecosystem function. So these results you can observe here also agree on what we observed from the fungal analysis that hinted at notorious antagonic roles of our fungal community based on the culture-independent approach. And lastly, in terms of the directed biochemical study, we observed that the extracts of these three species led to the identification of 7 different compounds with antibacterial, antiparasitic, and cytotoxic activities, which are promising against leukemia, breast cancer, colon cancer, hepatocellular cancer, and melanoma. So, as I already mentioned, these results into a notorious bioprospecting potential. However, none of these molecules are novel to science, so future works should implement the one strain, several compounds approach to trigger the expression of silent gene clusters, perhaps by using heavy metal stress. And with that, I'd like to wrap up some conclusions. So I hope that after this presentation, you agree with me that hydrothermal vents and oxygen minimum zones are hyper-diverse extreme systems for fungi, and that these organisms are key players in ocean biochemical cycles. And they act at different trophic levels, but their interaction with bacteria are very important. These bioresources are potentially useful for the bioremediation of marine oil spills and targeting those that reach the deep sea. The strong antibacterial activity against a set of human microbial pathogens highlighted the occurrence of some species like Aspergillus septi that targeted several pathogens. And lastly, well, of course, future investigations in bioprospecting should benefit from these stress-driven strategies to enhance the discovery of novel molecules. And with that, I'd like to thank all of my students, my research group here, and funding sources, collaborators. Once again, thank you very much, the organizers of this seminar, for the opportunity, and thank you for sticking around almost to the end. And with that, I'll be glad to take any questions.
Thank you very much, Dr. Vélez. Can you hear me? Yeah.
Yeah.
Thank you very much for the wonderful presentation. I got excited when you talked about hydrocarbons, bioremediation of it, actually. Thank you very much. I will let the audience ask questions if they have. Ladies and gentlemen, if you have questions, please raise your hand. Sachin Rajput, you can ask a question after 10 seconds. Okay, let me unmute him. Okay, Sachin Rajput, please.
Ma'am, what were those enzymes that were responsible for degradation of those crude oil and all those different components of heavier, mild, and lighter oil? Did you had study on them?
Well, thank you. That's a very interesting and very good question. We're actually, that's work in progress. I working on the identification of the enzymes, but well, from the literature, we know that the enzymes that fungi usually use to degrade hydrocarbons are those responsible of lignin degradation. So that might be my hypothesis.
And do you think that fungus could be a potential risk to oil reserves like oil wells or so?
Well, I'm not sure because the abundance of these organisms is relatively low. So I don't believe that they are a risk for the petroleum industry.
Thank you, Dr. Samir Damre, please.
Yeah, Dr. Patricia, wonderful work. Yeah, I just wanted to know, did you come across any barrow file as such during your isolation or other work? A barophilic fungi.
Yeah. Well, the isolates I presented in this presentation, sorry, these isolates, no, they are not typically piensophilic fungi, you know, those that are adapted to high hydrostatic pressures. But still, we have not done the experiments in the laboratory to observe differences or adaptation to these high hydrostatic conditions. So, of course, this should be very interesting, a very interesting experiment to do. But well, you know, we in Mexico, we lack the infrastructure to do these experiments. So
okay, yeah. So you haven't done any pressure cultivations as such?
No, not yet.
Yeah. Okay, thank you.
Yeah.
Dr. Mohan Kumar, please.
Yeah, ma'am, this is regarding when you were talking about the antimicrobial activity. Have you come across anything related to anti-biofouling property of the fungi which is isolated? Because most of the times the sheep and everything, it will have a fouling effect, biofouling. So anti-biofouling, anything have you come across?
No, well, that's also something we have not tested yet. Well, these are of fresh from the oven results. So, well, several experiments remain to be done. We have not tested none of those aspects yet. Sorry if this answer disappoints you.
Oh, that's okay. Thank you. Thank you. Dr. Mohan, please. Okay, Dr. Devdatta. I think Dr. Mohan Kumar has already asked the question. Dr. Devdatta, please.
Wonderful talk, ma'am. I would like to know, have you encountered any obligate marine fungi in your studies? like most of them are like seems to be like facultative or marine derived. So zoophila I have found.
Yeah, well, as you already mentioned, 97% of our isolates are marine derived, typical terrestrial fungi. But the Aspergillus sidowi is considered true marine fungus. And you can see this species in the marine fungal database is listed and is known for its association with, as pathogen with several marine organisms and affecting corals. It is related to this whitening disease in the Caribbean populations.
Thank you.
I have a follow-up question, Dr. Vélez. Anything what we see like penicillium or Aspergillus, we just try to dump them as marine derived fungi. But you find them in deep sea conditions. Deep sea conditions are the real marine conditions. I don't know these class. Bottle pigeon pigeon kind of what you call. This kind of a classification is probably appropriate at this time now. What are your comments?
Yeah, I agree with your observation. And actually, sorry if I mispronounced the name of this researcher, but there are several works on the adaptation of some Aspergillus terreus strains by the Rakukukmar. They obtained these isolates from the deep sea, and they grew them under high hydrostatic pressures, and they demonstrated the ability of this fungi to adapt to deep sea conditions. I mean, the high hydrostatic pressure. And well, this is has been repeatedly observed all around the world, India basin, the Pacific Ocean, Gulf of Mexico. I'm sure that these organisms might be also considered as a true marine fungi as well. But the definition, it's not helping. So we need to rethink the definition of a marine fungus.
I tell you because I am getting lots of interesting stuff from seawater from the beach. But they cannot really fit into your strict definition of marine fungi, and you put them as marine derived fungi, probably justified in some sense. Thank you very much. I think we last question from Mr. Vishal. And if you didn't know, Dr. Samir Damre and Dr. Varda Damre, they worked with Raghu Kumar, Sheshagiri Raghu Kumar, and Dr. Chandralata Raghu Kumar. They were from the same lab.
Hello.
Yeah.
Hello.
Yes, please.
Sir, ma'am, as you told about this bioremediation using this marine fungi, have you ever tried using nanoparticles made up of this marine fungi for this bioremediation process?
Yeah, well, no, not yet. Well, something I did not show today is bioremediation of heavy metals. And in this area, we're looking into, we're going towards nanoparticles, but not included in this presentation. And we are using a different set of isolates. But yeah, sure, fungi are very promising prospects for the production of nanoparticles, and they are related, well, in our experience, with heavy metals and calcite.
Okay.
The last comment from Dr. Rohit Sharma, please.
Thank you, Dr. Shenoy. Thank you, Dr. Patricia, for a very nice, informative talk, and also taking the questions and answering them patiently. I think your results of the fungi growing on carbon source on hydrocarbons is I think promising results for the bioremediation potential kind of thing. And very nice, informative talk. I think in future, your future research, we need to follow your future research, and we'll be awaiting your future research papers and articles. Thank you so much. Thank you so much for joining.
Thank you very much.
And thank you very much, Dr. Vélez, for spending. Wish you a great Sunday there, actually. Thank you. Ladies and gentlemen, we will now move on to the last presentation of this international seminar on marine fungi. The last speaker is our good friend, Dr. Samir Damre. He is another guru of deep sea fungi. You heard it from Dr. Vélez. She is one of the gurus of deep sea fungi. There are very few gurus of deep sea fungi. So you heard from Dr. Patricia Vélez. Now it's time to listen to another guru, Dr. Samir Damre. Dr. Samir, can you hear us?
Yes, very clear.
Oh, great. Before I hand over this platform to Dr. Samir Damre, Dr. Rohit Sharma, can you please kindly introduce Dr. Samir Damre to today's audience?
Thank you, Dr. Samir, first of all, for waiting to the last, and we are going into the last lap of our today's seminar. So Dr. Samir is a principal scientist at CSIR, National Institute of Oceanography, specializing in marine microbiology and biotechnology. He has extensive experience in studying extremophiles, particularly those from deep sea environments. His research includes bioprospecting for novel bioactive compounds and understanding microbial ecology. Dr. Damre is skilled in various molecular biology techniques, contributing significantly to the field of marine biotechnology. His work is crucial for discovering new microbial resources and their applications.